Hello students, this is Dr. Shabana Begum, Associate Professor from the Department of Zoology, Maharani Science College for Women, Bangalore, presenting a lecture on the histological features of mammalian testis pertaining to Zoology, BSc 4th semester under the paper entitled Comparative Anatomy, Human Anatomy, Cell Biology and Histology. The content of this section would be to know the histology of testis, its outer coats of layers, then seminiferous tubules, Sertoli cells and Leydig cells. So the learning objective would be at the completion of the lesson, the students will be able to learn histological details of testis and also learn the different spermatogenic cells, then Sertoli cells and Leydig cells and their functions. So this is all about testis. So before explaining about the testis and its histology, let me give you some points for introduction of the male reproductive system, which consists of a pair of testes, the duct system, leading from them the penis and the accessory glands. Right? So as you all know, testes are also called as male primary sex organs that produces sperm, and hence they are also called as male gonads. These are found within the scrotum, and in fact scrotum provides a temperature slightly lower than that of the body as required for viable sperm production. Fine. The duct system comprises of the epididymis, which hugs the external surface of the testis and serves as a site for sperm maturation and storage. Next is the ductus deferens, you can see in the slide also, extending from epididymis to the urethra, propels sperm into the urethra by peristalsis during ejaculation. Its terminus fuses with the duct of seminal vesicle, forming the ejaculatory duct. The urethra extends from the urinary bladder to the tip of the penis, and it conducts semen and urine to the body exterior. While the accessory glands, they produce bulk of the semen, which contains fructose from the seminal vesicle, activating fluid from the prostate gland and mucus from bulbourethral glands. So you can start explaining now about the testes, which are also called as testicles. It is the male reproductive gland or gonad in all higher animals including humans. These are found within the scrotal sacs or scrotum. Testis is homologous to the female reproductive organ called the ovary. So within the testis are coiled masses of tubes called seminiferous tubules. These are responsible for producing the sperm cells through a process called spermatogenesis. And the functions of the testis are to produce both sperm and androgen, primarily the testosterone. I just said the word spermatogenesis, that is, the sperm cells are produced by process which are called as the spermatogenesis. What is this spermatogenesis? Although you have studied about it, maybe you have forgotten, but still I'll give a gist of it. You can just listen to me. So the formation of spermatozoa within the testis in male animals is called spermatogenesis, which occurs especially in the seminiferous tubules. So the precursor cells in the germinal epithelium lining the seminiferous tubules begin to multiply by mitosis and form spermatogonia. That's called as multiplication phase. So even before the animal is born, however, the production of spermatogonia is most significant from the onset of sexual maturity, that is puberty. So they give rise to huge number of spermatozoa continuously until the age of about 75. So the process of producing one mature spermatozoon takes up to 90 days. The spermatogonium, destined to form spermatozoa, migrates inward towards the lumen of the tubule and enters a growth phase, which results in the formation of a primary spermatocyte, which is diploid in nature. The primary spermatocyte then undergoes meiosis, that is the reduction division, that's called the first meiotic division, resulting in the formation of two secondary spermatocytes. So here itself the haploidy is maintained and the number of chromosomes in the secondary spermatocytes will be n, that is they contain haploid number of chromosomes. So each of these secondary spermatocytes undergoes the second meiotic division, which is equational or mitotic, and produce two spermatids, which are spherical once again. So by a series of changes, the spermatids then become transformed into spermatozoa. And the process here is called as spermiogenesis or spermateliosis, during which time they are attached to Sertoli cells. When mature, the spermatozoa pass from the seminiferous tubules into the epididymis for temporary storage. Fine. So you have to know even the process of spermatogenesis along with the testis. It is not connected to the syllabus because in the syllabus histological details of testis is asked, but since the word spermatogenesis is included here, I have just given the explanation of it. Fine. So this is the diagram of the testis along with the duct system, starting with the epididymis, which continues as the vas deferens, etc. And finally you can even see the testicular blood supply. So once again, so you can just mention the testis, also called as testicles, are the male gonads that produce sperms. It is also an endocrine organ which is capable of producing hormones such as testosterone. These are present in the scrotomycete and these are suspended by means of spermatic duct. So these testes actually are paired oval structures measuring about 5 cm in length, 2.5 cm wide and 3 cm thick and weighing about 10 to 15 grams. So externally the testis is covered by 3 layers. These are the outer tunica vaginalis. It is the outermost incomplete peritoneal covering made up of connective tissue and the epithelium, and it covers the anterior and lateral surface of the testis. The middle layer is called the tunica albuginea. Fine. It is the middle layer formed by means of white collagenous dense fibrous connective tissue capsule. Fine. The third one, that is the inner layer, is called tunica vasculosa, which is the innermost layer having a network of blood capillaries. That's why it's also called as the tunica vasculosa, vascularized layer, that is which is highly vascularized and it provides nourishment to the testis. It's a thin and membranous layer also you can mention. Apart from that, you can mention each testis is divided into about 200 to 300 testicular lobules by means of fibers, connective tissue fibers or septa from tunica albuginea. So the tunica albuginea at the posterior end of the testis is thickened to form a structure which is called mediastinum testis, from which the connective tissue septa radiate into the testis and divide the testis into several pyramidal lobules. So the bases of the pyramids faces outward, that is the periphery, and their apices at the mediastinum. And even I said that septa do not form complete partition. and the lobules meet each other in many places. And the testis is divided into about 200 to 300 lobules, and each lobule there are 1 to 4 convoluted tubules present, which are called as seminiferous tubules. So here in the diagram you can see the section of the testis very clearly stained in the hematoxylin and eosin stain, that is called differential staining. The two layers, that is tunica albuginea and vasculosa, are seen clearly in this section, and you can even see the cut section of a number of seminiferous tubules which may appear oval or elongated also. There are sections of the seminiferous tubules showing the lumen, which is surrounded by means of two types of cells, which I am going to explain later, and in between these seminiferous tubules there are cells which are called as the cells of Leydig. Fine. So this is the diagram which you have seen previously. It is little enlarged and shown here to just get the detailed picture of the histological structure of the testis. A part of it is enlarged. The labelings also remain same, which I just explained about the coverings, that is the tunica albuginea, then tunica vasculosa is seen very clearly, apart from which there are the cut sections of seminiferous tubules, in between which there is interstitial connective tissue showing the presence of cells of Leydig, also known as interstitial cells. There are septae also seen, septa, fine. These contain the fibers along with the cut section of blood capillaries. I just said there are 200 to 300 lobules in each testis, and in each lobule there are 1 to 4 convoluted tubules. These are called as seminiferous tubules that are closely packed in the stroma of interstitial connective tissue. What is meant by stroma? So stroma is nothing but the basic internal structural framework of the organ. Is called as the stroma, and this contains blood vessels and groups of interstitial cells, also known as Leydig cells or cells of Leydig. These are endocrine in function, producing testosterone hormone, which is required for the development of normal secondary sexual characters in males. The sperm are produced in the tubules by a process called spermatogenesis, which I just described about. Fine. So then what you have to learn is certain terms or certain words which may appear in the notes also. So here the word mediastinum is mentioned. You can mention mediastinum is a region of tissue which connects the rete testis, which connects to the rete testis, and this supports the blood vessels and lymphatic systems, and the ducts within the testis which transport the sperm. Certain terms like secondary tubules is also mentioned in the notes. So you can mention the secondary straight tubules are the ones which connect seminiferous tubules to the rete testis, facilitating sperm transport, and they cross from within partitions which separate seminiferous tubules inside the testis. Then rete testis, actually they open into a series of channels called rete testis. So that means the seminiferous tubules, they open into a series of channels called rete testis, and these facilitate the transport of sperm from the testis to the sperm transport ducts of the penis. So you can mention The straight tubules and retate testes are actually found in the posterior of the testis. The connective tissue of the tunica albuginea extend more deeply into the organ. This inward extension of connective tissue is called the mediastinum testis, and it contains network of anastomosing channels called the retate testis. So, retate testis are nothing but anastomosing channels, and the epithelium that lines these channels is generally cuboidal. And also the epithelial cells, they possess a single cilium. So, these seminiferous tubules, they open into the channels which are called as retate testis by a way of the straight tubules or tubuli recti, also they are called as tubuli recti. Fine, that is about retate testis, straight tubules and mediastinum. Next, let us learn about the main structures of the testis called seminiferous tubules. Fine. So, these seminiferous tubules, they are enclosed by a layer of connective tissue which is called as tunica propria, which is made up of fibroelastic connective tissue. This inner to this layer is the basement membrane, followed by the stratified epithelium, and there are about 4 to 8 layers of cells found in this region, and the epithelium is also called as germinal epithelium. That you have to mention here. These are the tubules which lie within the testis and are separated by partitions which are extensions of the tunica albuginea, that is the middle layer of the testis covering. These tubules, they house germ cells and are the site of spermatogenesis or sperm production. The partitions, they divide the testis into lobules, and these lobules, they contain the seminiferous tubules, which I have just discussed just now. And I even said that each lobule contains 1 to 4 seminiferous tubules, and each testis may contain up to 900 of these tubules. The tubules average 50 centimeter in length and are tightly coiled within the testis. A typical testis contains up to 800 meter of tightly coiled seminiferous tubules. So, the diagrams which are here are enlarged and shown in the next slide. You can have a look at them. So, here this particular slide will show you the sectional view of the testis apart from the anatomical structure of the testis also. So, the first diagram you can see the testis lying within the scrotal sacs. This is a small diagram over there. The second diagram or figure will show you the diagrammatic representation of the testis showing the layer, the middle layer called tunica albuginea. There are also septa found in between them, and there are lobules shown there, and they are called as seminiferous tubules within them. Fine. Then there is a structure which is called as retate testis, into which the seminiferous tubules open, and along with that the duct system here, which in fact leads from the testis, the epididymis is shown, along with which the ductus deferens can also be seen here. There are also blood vessels which can be seen right at the topmost region of this diagram. So, the third diagram here will give you the sectional view of testis. So, here there are cells which I just mentioned about the spermatogonia, spermatocytes, spermatids, spermatozoa, etc. That is, they are the spermogenic cells which are seen in various developmental stages. So, here you can mention the seminiferous tubules have two types of cells, that is the spermatogenic cells that form sperms, and the Sertoli cells, which are the supporting cells. This is the diagrammatic representation of the seminiferous tubules showing a number of structures. So first you can mention about the spermatogenic cells. The section shows actually in various stages of development, and there are cells which are called as the spermatogonial cells, which are diploid, and their diameter is about 12 micrometer, and they are called primitive germ cells. Fine. Then the cells called as primary spermatocytes are seen, which are also diploid, and their diameter is about 17 to 19 micron. There are cells which are called as secondary spermatocytes. Apart from that, these secondary spermatocytes, when they divide, they form cells which are called as spermatids, which are haploid in nature, and their diameter is just 9 micron. Finally, there are the spermatozoans or spermatids seen towards the lumen of the seminiferous tubules. Fine, these are the variety of cell types which are seen in the mature testis. So you can mention this spermatogonial cells, they are rounded or cuboidal. They rest on the basement membrane. They have vesicular nuclei, and their cells are also known as primordial sperm cells. Next to it are the primary spermatocytes that are larger cells, and their nuclei show dark clumps of chromatin. The secondary spermatocytes are above the primary spermatocytes. These are smaller sized. Above the secondary spermatocytes are the spermatids, which are spherical and are the smallest cells with vesicular nuclei. So these are all the various spermatogenic cells which are found within the seminiferous tubules. Fine. Apart from that, there are also cells which are called as Sertoli cells, about which I am going to explain little later. So here you can even mention this sperm producing tubules called seminiferous tubules is highly convoluted and long, and it is about 30 to 70 centimeter long and 200 micrometer in diameter. These are surrounded by connective tissue that is called tunica propria, and it consists of flattened or spindle shaped myoid cells, myoid, myoid cells. Fine, you can see in the slide the purplish colored myoid cells clearly. What are these myoid cells? They may ask for 1 mark about what are myoid cells. So these are contractile in nature and help spermatozoa to move through seminiferous tubules. So the tubule as such is lined by means of stratified cuboidal epithelium, which we have already discussed, and it contains 2 types of cells. I said the spermatogenic cells in different stages of development and the supporting cells of Sertoli. So all the details and their cells can be seen. You can just go through this labeling once again. So this is another very beautiful picture of the section of the testis showing the seminiferous tubules along with the cells of leading. Fine. This actually the section shows the cell types involved in spermatogenesis, and in fact it is seen in histological sections using hematoxylin and eosin stains. So you can find the same cells of spermatogenic nature, which I have explained in the previous slide, containing spermatogonia, primary spermatocyte, secondary spermatocyte, and the spermatids. And also towards the lumen one can find the spermatozoans with their head embedded in the Sertoli cells. So in between these 2 seminiferous tubules you can find the cells of Leydig in the interstitial connective tissue, which secrete the hormone called testosterone. So this is yet another diagrammatic representation of the slide showing spermatogenic cells along with the Sertoli cells. Fine. So you can find the Sertoli cells, which is highly columnar in nature, with vesicular elongated nucleus can be seen, about which I am going to explain later. Then also there are a number of cells starting from spermatogonium to the spermatids. That means they are in different developmental stages. Finally, there is the formation of spermatozoa from spermatids by the process of transformation, which is called as spermiogenesis. You can see the spherical spermatids are going to get metamorphosed or transformed into a highly motile elongated spermatozoans. That process is called as spermiogenesis, where there are a number of changes taking place in the nucleus. Then there is a formation of acrosome, centriole, etc. during the spermiogenesis process. So before explaining about the cells of Sertoli, I will explain about the Leydig cells first. These Leydig cells are also known as interstitial cells. Fine. These are also known as interstitial cells. So in the adult male, the soft connective tissues surrounding the seminiferous tubules, they contain interstitial cells of Leydig, and these cells are almost non-existent prior to the commencement of testicular testosterone production at the onset of puberty. You can mention here they lie in the stroma of connective tissue. These are also called as interstitial cells. Then you can say they are large, ovoid or polygonal in shape with a large eccentrically placed nucleus. Eccentrically placed nucleus. The cytoplasm appears pale and vacuolated as the lipid droplets are dissolved during the staining process. In the cytoplasm, there are pigment granules and crystalloids, which are made up of albuminous substances. I said the Leydig cells are endocrine in function and produce the male sex hormone called the testosterone and to some extent estrogen. So they say the primary function of testosterone is to produce the male sex hormone, that is testosterone, which is produced in the Leydig cells, is to stimulate spermatogenesis, sperm production, and support the development of immature spermatozoa. So these are the functions of this. Then some 95% of testosterone produced by the male body post puberty is produced in the Leydig cells. One more point you have to remember: men who do not produce enough testosterone in their testicles, they develop a condition called hypogonadism, that is testosterone deficiency. So this is the picture of interstitial cells. Once again you can get a clear picture of it. You can see it's again stained in hematoxylin and eosin stains. Differential staining it is also called as. The picture shows all the cells of spermatogenic development apart from the Leydig cells, which are also called as interstitial cells, present in the interstitial connective tissue. In fact, for explanation's sake, you mention these are the cells which occupy a space between the seminiferous tubules and consists of loose connective tissue within embedded within them is the interstitial cells of Leydig. And we have already mentioned these are endocrine part of testis and secrete testosterone, a little estrogen and a little estrogen. One you can find right in this particular section the lumen line by means of spermatids. Above the spermatids there are secondary spermatocytes, above which there will be primary spermatocytes. Above the spermatocytes, just below the basement membrane, are the spermatogonial cells seen. So next, now let us discuss about the supporting cells of Sertoli. They are also called as Sertoli cells, which are drawn here in sky blue color. You can find the wavy outline or irregular outline of the cells also in this particular section. And you all should know that apart from spermatogenic cells, the seminiferous tubules have several supporting cells. These are also called as sustanticular cells. So that word K/A in this sentence says that called as. It is called as sustanticular cells, and they are irregular shaped tall cells, that is they are columnar cells, and the base of the cells, they rest on a basement membrane, and the apex extends into the lumen. You can be seeing the sectional view. Their nuclei are oval and basally placed, and they appear pale. There are one or more nucleoli, and the nuclear membrane shows a unique longitudinal groove there, and there are also elongated mitochondria inside, and the cytoplasm also has lipid droplets, glycogen and spindle shaped crystalloids within it. So here you can mention the Sertoli cells are supporting and nourishing, and they are surrounded by spermatogenial cells at the base, followed by primary spermatocytes, then secondary spermatocytes, spermatids, etc. Then the sperms are clustered in groups at the apical region, fine, within their heads embedded into the Sertoli cells. So their heads are embedded into the Sertoli cells. About the Sertoli cells, I have already just mentioned that they are the tall columnar cells, and these cells, they have lateral processes which form a sort of tight junction, occluded junctions also it's called as. What are these tight junctions or occluded junctions? Actually, these tight junctions are areas where the plasma membrane of adjacent cells are fused. That's junction. So these fusion points of the two separate Sertoli cells are forming a junction, and that is a barrier like, and this is called as blood testis barrier, which is very important here because it protects the secondary spermatocyte, then spermatids as well as the spermatozoa from the immune attack. So functions of Sertoli cells you can mention. These are the cells, already we have given the information. They provide mechanical support. They even provide nutrition to spermatogenic cells. They form a structure which is called as the blood testis barrier, and secrete androgen binding protein, and also they synthesize anti-Mullerian hormone, and also they try to phagocytize the residual body, then degenerating spermatogenic cells. So this is all about the Sertoli cells. Along with that, I had just said that I will explain about the blood testis barrier and its function. Fine. So this blood testis barrier is formed between the two Sertoli cells. That means the lateral wall of the Sertoli cell, the plasma membrane of which, right, they fuse together and form a sort of junction which is called as the blood testis barrier. This prevents blood and other body fluids entering the testis. This is the main function of the blood testis barrier. You must be remembering about blood-brain barrier. Like that, here it is the blood testis barrier, which try to prevent the blood and other body fluids entering the testis. It allows only secretions from Sertoli cells, such as the androgen binding proteins, protein-rich fluids, etc., to enter the lumen of the seminiferous tubules. So in doing so, the blood testis barrier enables the testis to maintain a fluid balance conducive to sperm development. It also protects developing sperms from the body's immune system, which we just discussed, and which attacks the immature sperm if the blood testis barrier is compromised, that is, if it were not to be there, definitely the immune system would have attacked the immature sperms. Fine. So this is the last slide of the lecture. The next will be the references. You can just go through the references. If you get the textbooks, you can read the matter once again and understand. Fine. Thank you.